record to the cloud. Very good. You're on.
Okay, so Mark, why don't you launch your PowerPoint presentation and we'll go from there. And I'll introduce the topic.
It looks, I'm getting a note. Oh, here we go. Okay. At first it told me no. So, and where did my PowerPoint go? Got too many windows open. Okay. Everybody see my screen?
Yes, that's perfect, Mark. Thank you.
Yep.
So the purpose for this meeting is to give you folks some training on how we do water quality sampling. You know, we've been doing stream flow monitoring, and now we need to get going with water quality sampling, and it's not as easy as you might think. Mark, could we go to the next slide? So just take a look at our overall watershed monitoring program. It includes a lot more than water quality work. We do habitat and geomorphology work. We've done that now on 3 of the different tributary streams that come into the Quiddy, and we've done that with summer intern students. We tried to get to that 4th stream, but we haven't been able to pull together the funding to get that done. We do biological monitoring with Dr. Rebecca Urban and her students at Lebanon Valley College. Mostly they do it, but during the pandemic, Trout Unlimited and Quidapila Watershed Association volunteers have been doing the sampling. Dr. Urban and her students do the invertebrate identifications and the write-up. And tonight, we're going to talk about the water quality monitoring component, and that's being done by you folks that are on the line with us now. Okay, so next slide, Mark. So the water quality monitoring program, you know, out of those 3 different components we're talking about tonight, the water quality program, we do stream flow monitoring, we do water quality sampling, and we do special studies. Stream flow monitoring is already in progress and going strong. Water quality sampling, we have not started, but tonight's session and the session coming up on Friday afternoon will get us rolling with the water quality sampling. And then our special studies, I really feel like we need to do temperature profiles, dissolved oxygen profiles. We need to see where, if the temperature exceeds tolerance limits for trout, and if so, where and where not. And also dissolved oxygen swings, dissolved oxygen minima. And we have the equipment now to make that happen. And hopefully we'll do that with summer interns or perhaps college students in special projects. So tonight, stream flow water quality sampling. So Mark, next slide. And our sampling program is going to be explained to us by Mark Hoger. Mark, we've already seen him. He is the water program specialist for PA-DEP. And I have to say again, he's been a phenomenal help to getting this program rolling. So Mark, I'll turn it over to you and you can take it away.
Thank you, Kent. And I've been very pleased with everything that you guys have been doing here. It's been really impressive to see the effort that you guys have put into this, building this program and this monitoring plan. So I'm happy to be partnering with you guys and providing some assistance. So just a real quick introduction. I've been with the department for 9 years now. I'm with the monitoring section of water quality down in central office, and my main area that I'm in charge of is dealing with the type of equipment that will, if people are participating on Friday, the training on Friday, the water quality equipment, we'll talk about it briefly today, and then sondes that are left in stream over periods of time to record autonomously water quality, select water quality parameters. And those give us the opportunity of doing things like Kent indicated, to measure dissolved oxygen over the course of a day, how it changes, how temperature changes over the course of the day, and through time responds to different events and things like that. So that's kind of what I spend most of my time doing, but kind of also have a lot of other specialties kind of get pushed in a lot of different directions in the monitoring section. But anyway, let's get started with what we want to talk to about today. Kent put the slides together, so thank you, Kent, for doing that. That was a big help. So just a quick overview of what we're going to talk about today. We'll talk about the target constituents, what we're actually going to be, what our results are going to look like, what we're measuring, the equipment that we use, how we use the field, how we take field measurements. Again, that's the equipment that we use to get readings instantaneously, how we collect the sample to be submitted to the Bureau of Laboratories for analysis. So what we can't measure in the field, we'll submit water samples to the laboratory to get tested. How we sample, how we handle those samples, how we process those samples. There is more than just filling up some bottles that we need to do in order to get some of these tests done. And then how we get the samples submitted to the lab properly and the paperwork that goes with it. So as I kind of work through this, feel free to ask questions. I'm not going to be able to, I don't think, see like people raise their hands or whatever when I'm in screen share. So feel free to just unmute yourself and blurt something out. I will also kind of pause after each of these sections just to make sure everyone has a chance. I'll also do a little bit of, I'll switch this off and go to just my camera to do a little bit of a demonstration in the sample handling part. So broadly, what we're looking at is we're gonna be looking for some parameters that are just strictly field measurements. These are parameters that are going to be changing through time, and so they don't do well with doing a laboratory submission to get those measurements. So there's some that we measure directly in the field. Broadly, we'll be looking at nutrients, the amount of, so that nutrients being like nitrogen and phosphorus, sediment. So what is like suspended in the water column that plays an important role in once it's deposited and fills in and around the substrate, it can affect the aquatic insect communities and therefore up the food chain. So we want to understand how much sediment is moving through the system, major ions and metals. So what are the field measurements that we're doing? We're doing 4 main parameters, 3 of which are going to change considerably if we were to try to submit a lab sample. Those are temperature, pH, and dissolved oxygen. So we want to make sure that those are measured in the field, and we'll have a piece of equipment that is calibrated and able to do that for us. Specific conductance, we also will measure in the field, though that is a pretty stable measure, and we'll also get a laboratory readout of that. So again, this is not something that's being tested with filling a bottle. It's tested with a piece of equipment right there in the field. Nutrients, we have a whole suite of various nutrient measurements, nitrogen based and phosphorus based, total and dissolved. So these can be kind of teased apart and tell us different things about the conditions in a stream. And so we have a whole suite of nutrient tests that we are going to apply. These will be collected in a couple different bottles, one that we will filter in order to get the dissolved constituents, and one that will just be a total. Talk about the bottles and all that processing here in a second. The sediment is quantified as a total suspended solids. So they basically spin this down and weigh out the amount of solids that are contained in a certain quantity of water. Some of the major ions, this is not an inclusive list, I don't think, but just to give you kind of an idea of what we're looking for. And then the metals. And I also don't have here listed, we will be testing for dissolved metals as well. So this is a handful of the most important, the most common ones that we're looking at. We'll take dissolved measures as well. And there'll be a handful of others that get in the mix as well. Okay, so I'll stop there. Any questions on the targeted constituents, what we're testing for before we continue on to how we do it? Okay, let's jump into the how. So what will we need? So the first thing, in order to do those field measurements, we'll need what we call a multi-parameter sonde. Some you'll hear us call it a field meter. You'll me call it a field meter a lot of times. This is a piece of equipment that has various sensors on it, and we'll have a picture here in a second that will allow us to do that. So we'll need that piece of equipment. Every time we go out and use that equipment, we test the calibration and recalibrate those parameters, and that makes sure that that those readings are accurate. So our DEP is providing the equipment as well as the standards that we'll use to calibrate the equipment, and that's what a lot of what we'll be talking about on Friday. So there's a logbook. I'll show an example of that. And then there's various solutions that are used again to do the calibration. We have a separate thermometer that we use to test the temperature thermistor on the sonde. That thermometer is a certified thermometer from a national laboratory that we can use to compare to our multi-parameter sonde's temperature. Extra batteries and bottles. So in order to do all these tests, there is a lot of different bottles. This is a very comprehensive standard analysis code. You'll hear that or SAC being referred to. This is basically the suite of constituents that we use to identify the tests that we want run. And it's a very comprehensive one. The nice thing about using a comprehensive standard analysis code like this is that not only will it give us the ability to ask and answer questions specific to this project, but having a comprehensive standard analysis code will allow us to use this data for future efforts that DEP might undertake to ask various questions statewide or about this stream and others. So it gives us a more comprehensive look the more we collect a comprehensive data set at all the sites that we do. So whenever we have the funding, we try to collect a full suite of parameters just to enable us to have a greater picture and a better idea of what things are statewide. More utility, I guess, than just this project is the goal. So there are 9 bottles that will end up being submitted to the lab. So there are 2 500 milliliter plastic bottles that are for general chemistry. They'll be labeled as Gen Chem. And so as a reference, everything in the parentheses is the label that's going to go onto it. I'll talk in a second about the label further. Then we have 5 125 milliliter plastic bottles, HDPE. There'll be 2 that are for the nutrients, the nitrogen and phosphorus. One of them is for total nutrients. One of them is for dissolved nutrients. There's two of the smaller ones for metals. One for total metals, one for dissolved metals. And then there's the fifth one is for the dissolved general chemistry.
I'm sorry, I accidentally muted you, Mark. I was trying to unmute myself to ask a question and I accidentally muted you, so forgive me. Can you unmute yourself? I'm trying to see.
There, I got it. Here we go. Good.
Okay, sorry about
Okay.
that. And can I ask a quick question here?
Please, please.
I'm wondering if we also, there's a possibility of measuring sewage, animal waste, and also there's a concern up by Beck Creek about a benzene plume. And I wonder if there's a possibility of measuring for other things beyond what you have here, like bacteria, sewage, animal waste.
Yeah, so there certainly is an opportunity to add to this. We can talk about various constituents of interest, and we can potentially add to this. I can speak specifically about the bacteria recreational use. If we can look into the current status of recreational use on the Quittapahilla. I'm not familiar with it but it could be an additional thing that we do to submit some bacteria samples. The approach that we use to test for that is a little bit different, but it's certainly possible. But let's talk about other constituents of interest.
Thank you.
In the future, and we'll do that for sure.
Mike, the bacteria test requires sterile conditions in the field. And when we did it in USGS, Mark, we had to burn the forceps in order to sterilize them and use sterile equipment. It's not something that I think volunteers could do, but it's difficult. Let's put it that way.
We can discuss it. Can't.
Okay.
Let's discuss that later.
Thank you.
Yeah. The last two bottles are amber glass, small vials, and those are for the total organic carbon. And in a second, I'll take my screen off, or take my screen share off, and kind of show you an example of what everything looks like.
Can I ask a question?
Yeah, please.
So what's the difference between, for example, nitrogen and phosphorus versus dissolved nitrogen and phosphorus?
So it's just sampling what is dissolved in the water versus what is like bound to sediment. And oftentimes we are more interested in dissolved constituents, or potentially interested in dissolved constituents. It can be more informative. So we want to be able to separate those. But really it's what's bound to sediment and what's been dissolved.
Kristen, the dissolved form of the nutrients means that they're pretty readily available for algal growth, or for bacterial growth for that matter. And the total form, phosphorus sorbs strongly to sediment. So if there's a lot of sediment, you're going to have a lot of total phosphorus. So there's a big difference between dissolved and total. Dissolved, of course, how do we get the difference? We've run it through a filter paper. Mark will demonstrate that for you in a sec.
Yep, exactly. Slides aren't going here. There we go. So on each bottle, I moved this slide up because I like to label in advance of going out. I think it just makes life a lot easier when you're in the field, you have enough going on. So oftentimes I label my bottles at a time. And then when I'm out in the field, that's one less thing I have to do. And so I put this under the equipment section. But really, there's any number of things that people put on the bottles. Our laboratory only needs very basic information. And so you're going to have a collector number as an organization to submit to the Bureau of Labs. I'll have that later. And that's always going to be that same collector. It's a 4-digit number. And then you will assign every sample, and a sample is all 9 bottles together is a sample. So that captures all the various constituents. And that sample will get a 3-digit number to identify it. So you'll have a 4-digit number, and then a dash, and then a 3-digit number. And then you'll have that label that is back here in the parentheses. So beneath the numbers, you'll write Gen Chem, dissolved Gen Chem, etc. So that's going to let the lab know. Essentially, the most important thing is what happened to each bottle. So each bottle will have a different processing. So one bottle might just get filled up. Another bottle might get passed through a filter and then filled up. Some of those bottles, whether filtered or unfiltered, will need to be preserved with acids to make sure that the constituents don't degrade over time. So the labels let the lab know which bottles they should be running which tests on. Okay, any more questions on equipment? A lot of the questions about like the.
Did you say that?
Go ahead.
I was just going to ask, did you say that the labels will be pre-made before we go out in the field? Is that, did I understand you correctly? Or?
We write on the bottles with a Sharpie. I'll
Okay.
maybe I can stop sharing and so I have a sample set of bottles here. You see that? So it's just, I'm just writing on the bottle. It's mirrored, isn't it? That's funny. So just writing on there the collector number, and then so that's my collector number 0948, and then dash, and then a unique sequence number. In this case, I did 740, and then general chemistry, Gen Chem. So I have 9 bottles, and they all have the same 740. So the next sample, if I go to another site, I would write 741. Next site, I'd write 742. But all 9 bottles are going to have those same numbers until I go to the next site. But I'm just writing on the bottle before I go into the field, just to save myself a step in the field. You could do it in the field too.
Well, yeah, that's the reason. The reason I was asking was if you were to use one of your bottles and it got contaminated in the process and you needed to get another bottle, you can do that in the field then, right? You could just
Absolutely,
switch
yeah.
in the field.
Yep. Number one thing is be very cognizant of what you're doing. If there is a contamination in the process, be willing to recognize that and start over. So sometimes you drop a cap and it goes down, and depending on the conditions, you might be able to grab it, maybe not. And so always have spares. Yep. Okay. I might go back to screen sharing. Go. Oh, and jump back to the. Okay, so jumping back into, let me get that full. Okay. So again, these parameters change. If we were to submit a bottle and ask for a dissolved oxygen test, I'll talk in a minute about how we fill these bottles. We leave a little bit of space at the top, and that would influence our dissolved oxygen reading. Same thing with pH, that little bit of air at the top can impact the pH. You'll notice that they do give a pH reading on our laboratory results, and it's usually close, but not exact. And temperature is definitely going to change because we put all these bottles on ice. So these are important to measure in the field in order to capture what they are. How do we do that? It's a piece of equipment like you see here in the picture. There's various different models. The one here pictured is what we've loaned. A couple of those, I've given them to Kent. I believe there's an XO2 that is on loan to the effort as well. It's another YSI product, and then a purchase of an XO3, I think you said, right, Kent?
No, the XO3, yeah, that's correct. Let's just to give you folks an idea. This is what we're talking about. This is the sonde. It's a big beast, as you can see, and this is the thing that connects to it and gives you a readout. So it's quite a little armful, I guess you could say. But on Friday, we get to learn how to use these.
Yep. Hands-on training then. So there's the details. If you aren't aware, reach out to Kent if you need more information, but I think you guys probably all have that information already. And we'll talk more about these on Friday. I mentioned briefly earlier that we keep a log of all of our calibration efforts. This is an example of that calibration sheet. Details on what piece of equipment we're using, date, time, who did it, the various parameters that we're measuring, and how the calibration process went. We keep a log of this as a regulatory body. Number one, I would say number one, it's good practice. It helps you stay informed with what's going on with the equipment, helps you identify problems as they start coming up. But it also gives more weight to your data to show that you've got record of going through all the right steps. Just makes your data much more defensible. So very good practice to keep record of all of this. Okay, so next we're going to talk about how to take a water chemistry sample. It seems very straightforward. It seems kind of funny to talk about in detail how to fill a bottle of water and give it to a lab. But there are some very important steps in the process to make sure that we are not influencing the sample or contaminating the sample, or biasing the sample. So a number of things. I'm going to start with something that I didn't really have in the slides, and that is to understand where you want to be filling the bottles. Oops. So this goes to this fifth bullet here. So our standard location is to collect in the thaw wag. What's a thaw wag? Thaw wag is the deep main part of the channel, and mid depth. So reach down and fill at mid depth. Now I do want to say that we will not always collect at this point. This is the default, but we use that piece of equipment that Kent showed you, and that was in the picture there, to help us understand across the width of a stream, what is the water quality like? And what we'll do when we first show up at a site is we'll take that piece of equipment and we'll just make our way across the width of the stream, taking measurements. In something like the Quittapahilla, probably five different points. We'll do it on each bank, and then 25%, 50%, 75% of the way. And what that tells us is if there is any variation across the width that might exist, it can inform where we want to collect that sample. So what could cause variation? Well, say you have a tributary coming in a short distance upstream. Sometimes it takes a certain distance for that tributary to mix fully with the main channel. And a lot of times what you end up having for a few hundred yards or maybe even longer is a stripe down one side that is different water quality than the main channel. And if you had recent rain, it might take that smaller tributary and push that further out. And it may even reach close to the mid channel. We want to recognize that and understand what we're actually measuring. So the most important thing is to understand what you're measuring and do that and select your location first. So when you go into the stream and you're doing that cross section, the next thing you want to be aware of is that you're not disturbing anything. Well, you're not disturbing, or someone you might be with is not disturbing anything upstream. You don't want somebody walking upstream, or you don't want to walk upstream especially if it's kind of slow flow, and kick up a bunch of sediment and debris that you would then be including in your sample. If you were to walk upstream of where you are, make sure to give lots and lots of time for that to pass, or simply walk upstream further to fill your bottles. Whenever you're filling the bottles, you want to face upstream. And when you're filling the bottles, you want to do it at mid depth. You want to make sure you're not collecting surface film. So there's going to be a lot of floating things at the top of the water. And if you just kind of scooped the top of the stream, you would certainly bias your sample. And we don't want to do that. We want to get a representative sample. So in the same way, we're going to be conscious of where we are bank to bank. And if that's representative, we want to make sure that we get down into the middle of the depth to collect and fill our water there. So we do 3 rinses with the native water before filling. And to do that, we want to make sure we're also rinsing with that mid-depth water and not the water at the surface. So one way to do that is to leave the cap on and then to uncap underwater, fill the bottle part way, cap it underwater, lift it up. The other way is to, can you guys see me still if I'm on my screen?
Yeah. Yes.
So so if you don't have the cap on, is to just, when the bottle goes into the water, is to simply have it straight facing down when it goes in, and then invert it once it's under. So either way is acceptable with DEP. If you have a best practice you want to employ and have them cap it, that's a great way. I think it's easier to do it right if it is capped. But either way, whether it's capped or just straight upside down when it enters the water, you want to make sure you're not filling at the surface. So you'll rinse 3 times. So you'll fill, cap it, shake it, and then when I go to dump this, I don't want to dump this straight in front of me because I'm facing upstream. I want to dump it behind me and empty the bottle behind me. Let the water let the stream take that away, fill again, and so forth. And then the final one, you'll cap underwater, bring it up. If you want to fill about to the shoulder on all of the plastic bottles. So if you have it more full than that, you can empty a little bit once you bring it up. The ambers, these are burned clean. So they're already certified clean. So these you do not have to rinse. So these little glass vials, stick them underwater, take the cap off, put the cap, make sure you get all the air out, put the cap back on, and we'll be done with this until we process. Any questions on the seemingly super simple?
Yes, Mark, I have a question. Suppose we find that there is a difference cross-section, that maybe there is a tributary upstream, and so the water on the left bank is not the same quality as the water on the right bank. Then what do we do?
Yeah, so at that point, it's important to the number one thing is to know if there is a difference, and then the next question is what is the most appropriate thing for what you're trying to, the questions you're trying to ask. In some instances, our group will take multiple samples and try to get a sample on each influence, and that's certainly an approach that you guys can take, and identify what each sample is representing. Most of the time, we're interested in that main channel. In this case, the Quittapahilla Creek. If there's a small tributary coming down the side, we're probably interested in the main channel. So what we're looking for, and the main parameter that we're looking at when we're looking at variation is usually the specific conductance. And DEP defines a significant difference in a cross section like that as greater than 10% variation. So if you have less than 10% variation in your specific conductance, we would consider that a non-significant change. If you have greater than, that would be significant. And so really it's whatever the study purpose is, but I would guess that you're trying to represent the larger water body, the water channel.
Quittapahilla is a fairly small stream, but it's also slow moving and not a lot of turbulence.
Yep.
So I'm not sure that the mixing is going to be all that great from bank to bank.
Yeah.
We'll check it out though, and we'll see.
Yeah, you see that in the wide shallow stream. So just on another example on the Susquehanna River at Harrisburg, we do everything in triplicate there because we essentially have three rivers at Harrisburg. The Susquehanna River water quality at Harrisburg has the Juniata River influence on the west shore. The Juniata confluences with the Susquehanna about 9 miles upstream. The West Branch confluences with the main stem Susquehanna River about 40-ish miles or more upstream at Selinsgrove, Sunbury, and up there, that entire distance, the water quality stays in a unique zone that whole distance down. So all the way down to Harrisburg, we still have 3 distinct water quality zones for exactly the same reason like you're talking about, Kent. It's wide, it's shallow, and it doesn't have a lot of opportunity to push that water and mix it around. So certainly a possibility in a stream like the Quittapahilla.
When we did the stream flow, we had the velocity meters, which give us a good idea on depth. So what are we using to measure depth for these samples?
So we say mid depth. It's an estimate. So you're just reaching down to fill it. So
so knee high, you're going to mid shin or things like that, waist, whatever you're
you just roughly the mid depth. Yep. And if we don't have it, you're not going to be filling probably in elevated flow conditions. So you just be wading out and dipping it down to roughly the middle of the depth. The most important thing is to not fill at the surface. If you do that, you'll okay. So, if I keep this screen up, would that be better, or should I go to just, if I want to walk through this, Kent, should I just stop sharing the screen here?
I think you should go to your full screen of yourself, Mark, if you're going to do a demo.
Okay, so just real quickly, all these dissolved constituents, we have to filter 3 bottles. And so when we do that, we're going to filter through a filter. It's a very, very fine pore size filter. There'll be a various rinsing that goes on, and then we'll fill the bottles. But instead of just kind of going through these bullet points, I thought it would be more helpful to just kind of show you the process really quickly, so that it might maybe clicks a little bit better. So we'll stop sharing.
So Mike, how can Mark show his screen, show his picture full width of the screen?
Yeah, it depends on how you configure it individually on your laptop. I have it configured on mine, so it's filled. I'm not sure how the recording does. I'm hoping that it'll work on the recording. So it's a long-winded answer. I don't know, but I think it'll show up fine. I'm hoping.
Well, this will be just the first introduction to it. There'll certainly be more introduction to what the equipment that you have. So I said before, there was 9 bottles. There's 9 bottles that you're submitting to the lab. I have a 10th bottle every time I go out. This is a 1 liter bottle. You could use a couple 500s or whatever you guys have. This bottle is a bottle that I fill in addition to the other 9. And then I use this bottle to do my filtering. This is my water that I use for filtering. So rinse this the same way, 3 rinses, and then fill it up. Filters are very expensive. So the process that we go through is to use a filter holder like this. And then we add in a disc paper filter to that to do it. And then we can reuse this plastic holder the next time with a new paper filter. So this just twists and comes apart. And then we have a box of our paper filters. So looks there. Resolution on this isn't great, but what you have in here, I got a pair of forceps. It's got a blue paper. That's just a separator. I dispose of that. Beneath that, oops, is another blue. They gave me 2 blues in between that one. And then I have a white filter paper. White filter paper goes onto the mesh part of the filter holder. And then I put it back together and I clamp it. And this is my filter apparatus. And then we use a syringe to push everything through the filter. So I've got my pretend filled water here. So my process personally, we want to rinse everything that we are reusing with ultra pure water. So this is certified lab pure water. Personally, I do this at the end of my process rather than at the beginning of the next process. And the reason I like to do that is I don't want anything drying onto these. So I give these a thorough rinse with the ultra pure water at the end of the process. And then once I'm starting this new process, I'm going to rinse with the native water. So I start by rinsing the outside of my syringe, which I'm about to dip in. I don't want to contaminate with anything that's on the outside of my syringe. So I rinse the outside of my syringe, and then I'll dip my syringe in as little as I need to. I don't want to just dunk the whole thing because, my hands are touching this and there's any potential contamination. So I'm going to dip it in just a little bit. I'll pull up about 10 milliliters, 20 milliliters of water. Then I'll pull it up the rest of the way to fill it with air, shake it, and then squirt it out away from all of my equipment. So I'm rinsing the inside of the syringe by doing that. I'll do that 3 times. Everything's triple. So I'll do that 3 times, and then I'll fill my syringe all the way. Okay. Now that my syringe is filled all the way, I can attach it to my filter holder, filter holder. I didn't mention this earlier. When I'm down in the stream filling bottles, the 3 that I have labeled as dissolved nitrogen and phosphorus, dissolved gen chem, and dissolved metals, these are not getting filled down at the stream because I'm going to only put in filtered water into these. So you can leave these at the truck. And then all I'm going to do is I usually push about half of a syringe through the filter to rinse the filter. And then I will put in about 5 milliliters to 10 milliliters into the bottle. So this is filtered water, shake to rinse, dump it 3 times. So I'm doing the rinses on the bottle just like I did in the stream, but instead of using straight native water, I'm using filtered native water to do my rinses. Make sense? You don't want to rinse with something that you're not filling it with. So once I've rinsed the bottle 3 times, I will fill it up again to the shoulder. And do that with each of these bottles. Just some best practices as you do this. You're always looking for sources of contamination. If it's hot and you're sweaty, don't be dripping, don't be hanging over the bottle, dripping in the bottle, right? Make sure you understand, like, are your hands clean? We can provide gloves if you want to wear gloves, but don't be, sliming up this with like sunscreen or mud or whatever as you're dipping this in and out. Just be very conscious of being clean. USGS uses the term clean hands, dirty hands, I think. Kent, is that right? Clean hands,
Yes,
dirty
that's correct.
Yep. So
So we have one person on hand to do the dirty work and one person to do the clean work.
yeah. So, the person that's actually doing the filtering probably isn't the guy that, I don't know, is over handling the sonde. And
Mark, we also
all that stuff.
wear multiple gloves, and when your gloves get dirty, you take them off, and there's another layer of clean underneath. So that's another approach.
Yeah.
If there's only one person working in the field.
Yep. And there's, the main thing is to be conscious of that. And then we also, we haven't talked about this a lot, Kent, I don't think, and it maybe isn't something we need to get into today, but we do a series of samples where we go through the process with that ultra pure water, or we do a duplicate do this exact same thing twice in a row. And those are quality assurance steps that we take to measure our ability to walk through this process without contamination. So we'll take some steps to ensure that everyone's doing this in a good way. But I think we all generally know some of those best practices things. So other things, whenever I have this, where I set this bottle matters to me. I don't want to set this in a place where I'm going to constantly be reaching over the bottles that I'm filling and working with. So I'll set this bottle off to the side so I'm not reaching over this and then potentially having my syringe dripping into my sample bottle, things like that. Any other questions or things that you want to add to the filter process, Kent?
No, that's good explanation, Mark. We'll have to just do it, guys, because it takes some practice.
That syringe, how many times do you need to fill that bottle?
So this is a 60 milliliter syringe and these are 125. So two syringes will fill a bottle.
So you are, I forget the term used, rinsing, contaminating that each time you get a syringe full before you put it in that bottle? Or just the first time?
So I'm only rinsing this the first time. Yep. After that, it's just keep continually getting more rinsed every time I fill it up, essentially, right? So rinse this the first time, and then after that, you're just filling it. So I'm just
Mark,
taking
what
it
is
off.
the cleaning process for the filtration apparatus and the syringe between sample locations?
Yep. So when I'm done, so I'll leave this together until I have preserved my samples with the acids and I have them safely in a cooler. Because what you'll realize is as soon as you disassemble this before that process, that's when you knock a bottle over, spill it, or mess it up, contaminate it. Now you got to put another filter in and go through that process again. So leave this all together till the sample is safely in your cooler. And then what we'll do is I have a bottle of ultra pure water. And I will rinse down the whole syringe very thoroughly. This is my squirt bottle of ultra pure water. And then I have it in a case, it goes in the case till next time. Filter paper comes out. And I thoroughly rinse this down with the squirt bottle. And that's it. There are certainly more involved precautions that other agencies or other folks do. This process for us has proven to be, we're able to consistently get good quality assurance samples. So when we do this process with ultra pure water, we come back with zero results. So zero contamination. So this is our process. There's certainly any number of steps that additional steps that you can take and other people do. But this is proven to be effective for us.
So for us, Mark, you can count on us submitting some quality control samples,
Yep.
equipment blanks, field blanks,
Yep.
and replicate samples. So all those will help us understand if we're doing a good job with our sampling.
Absolutely. Yep. And it's not always you. I mean, there's the potential for like your acids to be contaminated or something like that too. So we'll talk about that next. So once we've filtered, I guess I'll pull up. Well, I'm just gonna go through it while I have, instead of going back and forth, I'm just gonna just quickly talk about the fixatives, preserving a sample, and then I'll show the slide to kind of summarize. So some of the bottles need to be preserved. I mentioned that earlier in order for the constituents to not degrade before they are processed. So what needs what added to it? So anything that's general chemistry, we don't add anything to it. So the big gen chems aren't filtered at all. They're just filled. The little gen chem is filtered and then filled, but none of these get anything else added to them. It's just the water that goes into them. The metals, both our total metals and our dissolved metals, they will get nitric acid, and that will fix the metals for a really long time. They have a very long holding time once they're fixed. So what we do is we have a bottle of nitric acid, and we will take a pipette. And I'm sure that this isn't going to show up on the screen, but they are graduated. They've got little lines going up them. And we use those and we put 2 milliliters, basically 1 pipette full of acid into the bottle. And that will bring down our samples to below a pH of 2, which will fix those metals in place. So each of these bottles gets 2 milliliters of nitric acid. Our nutrients dissolved and total. And these vials for the total organic carbon, TOC, they all get sulfuric acid. Obviously don't want to use nitric acid, which has nitrogen in it, to add that to our nitrogen tests. So that's the way I remember which one goes in which. Not adding nitrogen to our nitrogen samples. So these bottles also get 2 milliliters of the sulfuric. So the nitrogen and phosphorus get 2 milliliters of sulfuric. These little guys are going to get about a half or so milliliter. And what we do with these is we don't, so these bottles, we want headspace, so we fill to the shoulder. These we want no air in them at all. So you will, once you uncap this, the water will be right at the surface, and we'll just add about a half a milliliter will be just enough to kind of give you a little bubble of liquid at the top. The acid will all be sinking towards the bottom, so you don't have to worry about when it kind of drips out. It's not the acid is all gone to the bottom because it's heavier. And you'll cap this, and when you cap this, you'll see a little air bubble, and as you cap it tighter and tighter, the air bubble will just go away. So, and that's all you have to do with those. So that is how we preserve them. And then all of these bottles go on ice until they get to the laboratory to help, again, hold them. So I'm going to go back to my screen share. Any questions on the process of preserving the samples? There it is.
So how many coolers do you need in your truck to hold all this stuff?
Yeah, depends on how many sites you're doing and how big your coolers are. So we have some decent sized coolers. If you guys are in need of coolers for your effort, let us know. We can certainly loan you a cooler or two. We also, I've talked to Kent about this, in order to save on cost with ice. We use frozen water bottles or other plastic bottles that we freeze. So if you got a little bit of freezer space that you can freeze some water bottles to use to put on top and around, that's another good way of doing that. But you might benefit from having some decent sized coolers, because these 9 bottles will take up a good chunk of space for one site. So we don't need to go through this. It's just a summary of what I just said that you guys can have as a reference to go back. I think Kent's got this all documented in his documentation as well. But there you go. Okay, so how are we doing on time? We're almost done. So next step is to just get it to the lab. Down here, I've got the address. It's off the Progress exit off 81. Though I need to confirm this still. I'm pretty sure this is the case. The best approach is to get there at one of these times. So all samples have to start being processed the next day for some of these tests. So samples need to be delivered either that afternoon to the Bureau of Labs, or you can get there around 7:30 in the morning, the next morning, when they're starting to grab those samples. The process is basically that there's a little phone by the sample receiving door. You can call over to the secretary and someone will come let you in, and you can there's a large refrigerator that you can put the samples in. It'd probably be best to meet you guys at some point over there and kind of walk you through that process. But so I won't go into any more details for now, but you do want to make sure that they get there the next day. So for that reason, also you'll need to collect samples where the lab is open the next day. So generally that's collecting samples Monday through Thursday. Because if you collect a Friday sample, they won't get a sample on Friday, they won't get to it till Monday. Obviously there's some holidays that interrupt that as well, but they need to be open the next day. With your samples, you give them a piece of paper that let them know what's going on. This is an example of that piece of paper. Kind of run through some of these pieces on the next slide. What I'm going to talk about on this slide is this. It's really small, but this is the section where you tell them which bottles to be receiving. These unpreserved, unfiltered are your Gen Chem. So I have 2 samples in this example. I'm submitting 2 samples to the lab. So these are the big bottles. So I have 2 samples times 2 is 4, and so forth. So you're just filling in all of this, how many bottles they should receive. I'm going to go to the next slide and just kind of talk about these different. So this is what's being filled in on the laboratory submission sheet. So the Quittapahilla watershed group, it has this collector identification number for the whole group. So any sample that's collected for this effort would go under the collector identification number of 4479. So when I showed the bottle, I have my number is 0948. Every sample that you guys will start with the 4479. And then after that is the sequence number as well. So that's the second part. That's the unique 3-digit number that you're adding onto that. So there's 3 codes that classify your sample and tell who to charge and all that. That's these at the top. So probably the easiest thing for you guys is to just have the collector ID number, reason code, cost center, and program code. You could have these all populated and then just make copies of it with those in, so you don't have to remember those codes. You can probably enter some of this other stuff too. But for sure, those could be entered up front. Date collected is pretty straightforward. Time collected seems straightforward, but we make it a little bit more complicated. First of all, we do 24-hour time, but more confusingly, we do not recognize daylight savings time on our samples. So right now we're under daylight savings time. So we use Eastern Standard Time, which right now is 1 hour behind our current watch time. So put Eastern Standard Time for all of your samples and whenever you're recording data. So right now it's 9:06. So if I collected a sample at this point, I would be putting a timestamp of 8:06. Actually, I'd be doing a timestamp of 20:06 for the 24-hour time. Okay, sampling location is not necessary, but it does help keep things a little bit more organized. And Kent, you mentioned, how do we are getting all this information into the database, and I said that I will be the person doing that. One way to help communicate that can be that you guys can take a picture of the sample submission sheet and then forward that on to me. And if you put the sampling location in here, just a quick description, that should be able to help me get the locational information tied to the sample in the database. So if you take a picture of, if you do fill out sampling location and take a picture to send it to me, that might be a helpful workflow.
Okay, so Mark, question. Now, if you go back one more slide, back to the bottles, where we have, what are we supposed to put on the bottle? we're supposed to
Oh.
put in,
Here.
yep, exactly. Collector number, sequence number, and the SAC. So
I'm
that
sorry.
is all tied into that sample submission sheet. And once you have the sample submission sheet in your hand, then you know everything you
Yep.
need to know. So
Yep.
now go back to the sample submission sheet. And you see you've got there the sequence number and the code and everything. And so you know what samples were submitted. So Mark, do we get the sample submission sheet returned to us from the lab, or do we need to make a copy of that?
So you will not get this back from the lab. So what I mean, I make notes in a field journal of all this information, and that's the main thing I go off of. Some people get in the habit of taking a photo or making a copy of this every time. I'm not one that does that unless I'm trying to coordinate with somebody else, and then I will do that. If it's just my own project, I won't take copies of this. But it can be something that is a helpful record, especially if you're not keeping a journal, which I would highly recommend that you do. Have a notebook that keeps a log of this stuff. If nothing else, you're going to need a place to write down the field meter readings as well. So that's the other piece of information that I would need from you guys. In addition to the information on this sheet is the field meter readings that are associated with the same location, same sample. This standard analysis code, the 087, this tells the lab everything that they need to do with these bottles. So in addition to the tests that they need to run, it tells them how precise they need to be for each of those tests. So some organizations just need, like in the mining program, let's say they're doing a lot of tests with like tons of heavy metal issues, right? They want to know is our iron below 200 micrograms per liter, but other programs like us, we're going to want to know, have a lot more fine level test for those lower levels of contamination. So it communicates all of that. In this code, this is an established set of tests to run and exactly how to do it. It's much easier than writing out all of that stuff every time. So it's just a way of communicating. Yeah, I think that pretty much hits it. Yeah, and the last thing is you're just signing and dating and time that was delivered, and you're all going to be doing hand delivery to the lab. So, and contact information if they don't see all the bottles or there seems to be an issue, who to contact. Okay. Any other questions on that? Here we go. There's the standard analysis code again. So
Is rainfall a contamination issue if you're collecting?
yeah, number one, you don't want it to be raining into your bottles. So if you're doing the processing and all that, try to get into a dry area to do your filtering as best you can. As far as like having runoff into the stream, I think it's important to just note that and to be aware of whether or not you use that in your analysis might be something for consideration. So when you have that runoff and your stream turns to chocolate milk, or sometimes even if it doesn't look that bad, you're going to have very high levels of a lot of these constituents as you have an increased suspended sediment and all the things that are attached to that, whether it be metals, nutrients, etc. And so if you are sampling during a rainstorm or shortly after a rainstorm and you have differing flow conditions, it's important to make notes along those lines We can also kind of understand some of those things by looking at the data, right? So as you get used to looking at the results, total suspended solids is abnormally high. Well, either somebody was stomping upstream or you contaminated your sample in some way, or you sampled during a storm event. So you can kind of understand a little bit of maybe what was going on. But yeah, sometimes it's important to target those events too. You know, various efforts will be made to target those storm events to understand how much contamination is running off in the stream. So it's all part of your project goals.
So question to Kent then is try and sample at various times or different events. Is that what you're saying, Kent?
You're muted, Kent.
Yes, Gary, we want to, can you hear me now? Yeah.
Yes.
We want to take a variety of different environmental conditions as best we can. But if it's raining, try to protect your sample. But if the stream is muddy, we go ahead and do the collection. Because that tells us a lot. It tells us what happens during high flows.
Majority of contamination in a stream takes place during those events. Okay. Just a summary of what we're doing. So when you guys are collecting all of these measurements in the field, make sure you're noting them down, and all of these will get gathered and provided to a DEP representative to get in the database. And for now,
That first, Mark, just to step in here a minute, that first number one there, field data collection sheet for discharge measurements.
Yep.
That's this thing that we've already done in the field when we do our stream discharges. And the second one, field meter calibration form, we'll learn about that on Friday. Mark mentioned that earlier, but we'll do that on Friday when Mark shows us how to use the multiple SOND. And the multiple sample submission sheet is what Mark just went over. So there'll be those three documents. Now, Mark indicates that we need to do a field journal, and certainly we will do that, and that makes a lot of sense. So that'll be a fourth item.
do you want to jump in and talk about this then, Kent?
Yes, I do. Everybody, we've seen this a number of times already, but water quality changes as stream flow changes. In general, suspended substances increase whenever the stream flow increases. And in general, dissolved substances decrease when the stream flow decreases. So that's why we want to do a stream flow measurement every time we collect a water quality sample. Now I've drilled this into you folks, but one more time, and probably this won't be the last time. Go ahead, Mark.
Yeah, I think it's great. Definitely. Here's a checklist. You said
So
you've done
that's just a
something this already?
checklist for when we go into the creek with our stream discharge measurements. We no need to go over that now.
Checklists are fantastic. Glad to see you got them, because as soon as you don't use a checklist, that's when you are making extra trips. Okay. That's the end of it.
Great, thank you, Mark. Do we
Any-
have questions?
Yeah.
Are we
It's a lot.
overwhelmed?
Yeah, it's a lot to go through with a PowerPoint and listen to someone yap. It'll all make a lot more sense when you get in a stream and you can actually do this. The more you do it, I mean, gosh, you do several of these, and before you know it, you can do it with your eyes closed. But
Automatic, right, Mark?
yeah. And
No.
I think, well, I think it. I mean, it does get that way. I mean, it may take a while, but I think the main, like my last piece of advice is to get in a habit of doing things the same way so that muscle memory does take over to some extent. Whenever I'm filtering and doing my preservation, my bottles are always in the same order every time. I always have my metals over here, and I have my nutrients over here, and that allows me to do things just kind of stepwise and you just, like I said, muscle memory, do things the same way, and you'll start to form those good habits of sample collection.
Questions for Mark?
Well, I have a question for both of you. When we did stream measurements, the velocity, we were a little disappointed of, some places we had eddies and, we discussed that. Do we have a project, or do we have to look for better sites along the creeks that we're measuring to get that velocity that you feel
Good question,
better about it?
Willie. Mark, we did some stream flow measurements in Killinger Creek, which is a tributary to the Quittapahilla, and in Beck Creek, which is another trib. And the Beck Creek site was influenced by muck and weeds, and the Killinger site influenced by weeds. And so the velocity at the surface was much faster than the velocity where we were doing our velocity, where we had our velocity meter positioned. So Willie, I talked with my former colleagues at USGS, and they said a couple of things we can do. We can do the velocity measurements at 2 points at 8/10 the depth of the creek and 2/10 depth, and average the two, or we can try to find a better spot, or we can even quote manicure the site that we do a little bit more. In other words, clear out the weeds farther upstream, a longer stretch, so that the flow is not influenced by that weed patch. So yeah, we took some stream flow measurements that were not acceptable. We wasted our time there, but now we know, so we'll do better.
It can be very tricky in streams that, well, in small streams to begin with, streams with vegetation and other mucky bottoms and things like that. It absolutely is a challenge. Yeah.
I guess the key is to not disturb it upstream of where you're going to collect the sample, right? So you don't want any sediments or anything kicking up.
In the case of this, yes. With the velocity meter, it doesn't matter.
It doesn't matter.
Yeah.
Yeah.
The other, one last thing to just mention with taking those stream flows is do all that stomping and clearing before you start. You don't want to do half of your measurements and then go stomp another section out a little bit or move any rocks. I'm sure you've talked about that with them, Kent. Do all of that adjustment to the environment prior to you starting, because as soon as you move a big rock or eliminate some weeds, that changes. It could change the flow measurements and all the water's being directed to that where you just cleared out as opposed to where you just took measurements. So it can affect your reading overall. So do all of that clearing up front and then start and do all your readings without adjusting anything.
Any other questions?
Again, my sense is it's as you were saying, Mark, is the kind of thing you just have to do and just do in the fields. I look forward to doing this with y'all.
We'll have some fun, Mike. Hey, everybody, thank you for your attention. And Mark, especially thank you for your presentation and your time. We really appreciate it. So I'll give you one last chance. Bob, did you want to say something? Dave?
Thank you.
Thank you, Mark. Yeah.
All right, everybody. Thanks.
Yeah, thanks very much,
All right,
Mark.
thanks, everyone.
This will be posted on YouTube, and for the edification of the world about water monitoring, so.
How to
And
fill
Mark,
bottles.
Well, that's perfect.
and Mark, will
Thanks,
you
Mark.
send me the final PowerPoint, please? So that
Yes,
I can
I'll-
share that with everybody.
Yep, I'll send that to you, and I bet I'll be seeing a number of you guys on Friday. So
Absolutely.
take care.
Hey, good night, everybody.
Thank you.
Good night.